Hello, everyone. My name is Harry He, your host for today, and welcome to our webinar on innovative pathways in drug discovery, integrating DNA-encoded library DEL technology. Drug discovery is a complex, lengthy process involving the identification and testing of numerous compounds to find those with the most potential for new treatments. With the advent of new drug discovery technologies like DEL, there is a growing interest in innovative methods to accelerate this process. We're very excited to have you join us today and learn about this cutting-edge approach to drug discovery. So today we're joined by two experienced industry experts, Dr. Bing Shah and Dr. Wes Kazmierski, who will share their valuable insights on the future of drug discovery using DEL technology. We'll also give a 5-minute Q&A session at the end, and you also get a chance to directly communicate with our speakers. So please feel free to ask questions and type them in the Q&A chat box. So today, before we dive into the presentation, let's take a moment to get to know our esteemed speakers who will bring a wealth of knowledge to our virtual stage today. Dr. Bing Shah is an accomplished chemist with over 15 years of experience in DNA-encoded library DEL technology. He earned his PhD from Boston University, mentored by Professor Guilford Jones, and collaborated with Professor John Porco. He's a former GSK Associate Fellow, leading projects in multiple therapeutic areas. Dr. Shah has received numerous awards, including 3 Exceptional Science Awards, and was a finalist in the GSK 5K Electroceutical Ideation Challenge and the $1 million incentive. He's organized major conferences like the GSK 2019 Global Chemistry Conference and reviewed over 100 journal submissions. His work has led to over 20 publications in top journals. And Dr. Bing Shah is currently the Vice President of Viva Biotech. And today we'll also have the honor of hearing from Dr. Wes Kazmierski, who is a medicinal chemist and drug discovery scientist with many years of experience in academia and industry. He co-developed the one peptide, 1B library approach at Selectide Corp. He led many discovery and optimization programs at GSK and Viva Healthcare, delivering a number of clinical stage compounds, and was named a co-inventor of the drug Lexiva. He was selected a GSK Fellow, and in addition to many others, received 4 GSK Gold Awards for enabling clinical programs. He is currently a Senior Director at Biohaven Pharmaceuticals. His chemistry expertise includes small molecule receptor binders, enzyme inhibitors, protein degraders, peptides and mimetics, protein drugs, bifunctional antibodies, antibody-drug conjugates. His therapeutic experience includes virology, cancer, immunology, neurology, and autoimmune diseases. He co-authored more than 200 peer-reviewed publications and patents. So today, to ensure a better quality of this webinar, please keep your microphone muted during the presentation. All right, without further ado, I will now give the floor to our speakers, Dr. Shah, and please feel free to share your screen and begin your presentation. Thank you.
Great. Can you hear me well? Okay, cool. Thank you very much, Harry, for the kind introduction. Thank you very much for Viva and Viva's marketing group to put this together. It's a lot of backstage work. I really appreciate it. Last but not least, of course, thank you very much for Dr. Wes to agree with me to have this this chance to review those exciting science again. Thank you very much. Okay, so I'll go over for the presentation. I'm kind of lazy. I just borrow the the advertisement of this agenda as my title slide. The next slide, please. So we have a little bit modification of our today's agenda. Instead of 2 separate presentations, Wes and I are going to give a tech talk. So pretty much this tech talk has 3 parts. For those audience who are not so familiar with DNA-encoded library, I will give a brief introduction of DEL, then followed by a case study of ID1. Hopefully, this is a part for give some flavor taste for somebody already been a DEL users and expert in medchem. Then we'll close our presentations with the conclusions, insights, and some also from both sides as DEL providers and the DEL users. And also maybe we'll share some even personal opinions of this technology and the drug discovery platforms. Next slide, please. Okay, introduction of DNA-Encoded Library. Next. Okay. So for those not so familiar with DAO, so I always like to use this drug discovery pipeline scaffold to introduce where the home for DAO in drug discovery traditionally. Pretty much DAO, the home of DAO is belong right after target discovery. It belongs to hit finding and assessment. We have a golden standard of this part is high throughput screening. And normally I like to use compare with HTS to DAO to help people understand what exactly DAO is. Next slide, please. When I trying to introduce DAO, I always like to use this essay news cover story to tease audience a little bit. As you can see from here, Professor Konerger from Stanford University, he's also a 2006 Nobel Prize winner in chemistry. He made a very, very strong statement here. He said, DNA-Encoded Library are revolutionary. It represents the most innovative and significant advance in chemistry in the past decades or so. It's not a most, it's not one of most. As a chemist, even I funded it a little bit. Next slide, please. Also, I like to use a Nature publication. Retrospectively, we find its prediction is pretty accurate. At that time, they already predicted that DNA-Encoded Library are set to become one of the pillars of discovery in the pharmaceutical industry. Nowadays, yes, there is pretty much every big pharma or small biotech either have a small in-house team or some point person to collaborate with their providers. Next slide, please. Okay, enough for the teasing. So what is DAO? Actually, DAO is not as novel as we thought before. Actually, it is a thought experiment proposed by Berner and Lerner as early as in 1992. So the idea is actually trying to do a dream at that time is also we can summarize the whole concept of the DNA-Encoded Library into one sentence is to screen billions of compounds in a single tube. How are we going to do it? The basic fundamental idea is you put the DNA barcode of each small molecule. And in that way, you can do that. And also that is revolutionary part compared to HTS. The HTS is the experimental per vial concept or cost per vial concept. Once you screen billion compound in one single tube, as you can imagine, the cost, the time will significantly reduced. Next slide, please. Okay, so the thought is there, the idea is there. So how we're going to realize it? Actually, we have many different approaches now. We have DNA record chemistry, we have DNA template chemistry, we have, it's the self-assembly ones, and lately we also have 1B1 compound DAO library technologies, all kinds of this version. But today, I'm gonna only laser focus on the first one, DNA record chemistry, because this is very popular, or actually it's dominating across industry. GSK, Viva, the Wuxi, Heijin all use this platform. The reason actually is because this methodology is reliable and straightforward. Next slide, please. Okay, so for this type or this version of DAO, this is the basic flow chart of fundamental of DAO. Don't worry, I will elaborate each step a little bit in the following slides. So pretty much for DAO, you can narrow down to this, we can see about 4 steps or 6 steps. Number 1, you have to make DAO libraries. Number 2, you will use the DAO libraries to apply for the affinity selection. Then after affinity selection, you do NGS sequencing, then transfer those for the data analysis. Because the DEL, the amount is so little, you have to resynthesize those off DNA, those compounds recommended by the data analysis, then do a hit evaluation in normal bioassay. It could be biophysical or biochemical. Once they confirmed, you find a hit. Next slide, please. So actually, I'm gonna elaborate each step a little bit more here. For the step one, how to make a DEL library. No matter how complex or how easy or how diverse of the DEL libraries, most DEL libraries, surprisingly, actually starting from single one thing, very identical thing almost, we call headpiece. This is a different version of the anatomy of the headpiece. As you can see, there's a left hand of the headpiece have a free amine. It served as a handle for chemistry. Then they covalently link to a short DNA duplex. And the short duplex have purpose to have two base overhang. That's is handle for the enzymatic library, ligation or encoding. Next slide, please. So let's focus on the left side of the slide first. So let's see the fate of one single DEL molecule. As you can see, actually, this DEL molecule is grow chemically from left side to get bigger and bigger, and grow enzymatically from right side get bigger and bigger. So okay, to jump into a little bit more details, we start from headpiece. Then the first step, you do encoding. For example, here, a yellow DNA barcode. Actually, the building blocks and this coding tags are already organized beforehand, like in Excel sheet something. You encode this first, then add a corresponding building blocks like yellow one. Then you add this building block 2 encoding, which is green. The building block 2 here we represent as a green as well. So that's the fate of one single molecule. And that's repeating myself again, grow two directions simultaneously, one chemically, one enzymatically. But how we make molecule actually is we use the so-called split and pulse synthesis strategy. That's on the right side of my slide. As you can see, we started from headpiece, then split into 96 well plate. So each plate, each well have we can individually do the ligation. After that, we'll individually do the chemistry, but the ligation DNA part and BB part are one-to-one relationship. So once it's done, we can pull them together, then split again. Now in this plate, each well have 96 times 96 unique compounds. And then if we do one more round, you get roughly 100 times 100 times 1 billion, 1 million diversity here. That's the way, that's the beauty of this split and pulse synthesis can grow this technology or grows the diversity of the library like crazy. Okay, next slide, please. So this is actually how we make the DEL library. This is how we do the affinity selection. This relatively straightforward. You immobilize the target with DEL, then some DEL will be binding to the target, some will floating around. Then you do a wash, get rid of the non-binders, so the supposedly strong binders will survive. Then you do a release by degradation of the protein. You detach those small molecules, which is supposed to be binder. You can do 3 rounds to give them even higher, competition pressure. Then in the end, you're gonna collect those strong binders, then do a PCR and the sequencing. This will tell you which small molecule on the other hand is binded to this target protein. Next slide, please. Okay, so because today we don't have enough time, I just deep dive to 2 parts. One is the how to make a DEL library, and 2, how to do DEL selection. But this already can tell you the key information or key advantages of this technology. Number 1, you can, in very short period of time, make ultra-large libraries with deep chemical space with SAR. As you can see, 1 million is very quick. And also, an advantage of the, you said, the affinity selection is, as you can see, compared to HTS, there's no need for upfront activity assay development. And as you can imagine, the amount of usage of protein is very minimum. And also we can do automatic parallel different conditions. So if you combine them together, actually you can immediately realize this is the perfect technology for first IC, first in class targets, because you don't have to know much about this target in beforehand. Next slide, please. Okay, from money perspective, that's also some advantage of this technology. This is not this arguably there's some way to calculate this. It's not a way calculated. It's natural publication there. Roughly, people think that they're gonna cost $1,000 per compound for a HTS, but much cheaper for a compound. But this is arguable the way. But generally speaking, this is a less expensive technology for sure. Next slide, please. Okay, so that is the very fundamental knowledge of DAO. Now we're gonna introduce a little bit of video. Anyway, this is the Viva program, but I won't spend too much on there because the real case study will get you even more excited. As a big believer and the practitioner of the DAO, actually, what the solution and the proposal from Viva, actually, we highly recommended the synergy between DAO screening and other hit ID methods, such as HTS, ASMS, FBDD. Because based on my experience and based on so many years, the data show that actually the synergy between different platforms give the highest success rate. Next slide, please. So the narratives or solution from Viva or VDAV, we think is actually when we tackle the targets, no matter first size, FIC or BIC, the order of the flow is going to be affinity first, specialty paves the way, then good molecules will come naturally. And this, you can see some of the right side of this slide. Pretty much the affinity first, the ASMS, SPR are going to take care of that part. The specificity, of course, some screening part, and also MedChem had to lead will take that part. Once we entwine them together, we'll have the good results. And also nowadays we put AI as a big supporting tool of every single step, not only DAO self, but also SPR, FBDD, ASMS. We don't have time to elaborate this part now, but keep tuned. We have more seminars about this. But in terms of DAO, AI, okay, that's fine. AI is, we will use AI to help each step already as well. Then put this in the bigger picture. So this is the kind of a summary of the Viva technology platform. What we propose again is a holistic approach. The two squares there, as you can see, we have an affinity-based screening platform and biophysical technologies. Those are strictly belongs to affinity first, but other things we can loosely belong to this as well, such as I just mentioned the AIDD, CIDD bioassay platform, protein production, structural biology, which is our super strength, and membrane protein technology, GPCRs, and also cryo-EM, even antibody DMPK. That definitely is part of the story, part of the solution. And surprisingly, if I ask some people not familiar with DAO, what is the bottleneck of this technology? Actually, the answer may be, yes, answer may be shocked to you. The bottleneck actually is the post-selection chemistry, almost goes to the MedChem part. The solution of the Viva is actually, we have many chemists. Now our manufacturing videos now in Shanghai and Chengdu. They bring out this more libraries day and night. Also, we use many legacy synthesis BBs to be part of our libraries. And the most important part I like to see, the PSC is readily available, which is very important for a DEL target. If people did that before, you will agree with me. Next slide, please. Okay, so this is a brief introduction. Let's jump into the case study. So I'll hand over this stage to Wes.
Yeah, thank you very much, Bing. Next slide, please. So first of all, I'd like to thank Bing and Viva for inviting me to be guest speaker here. I'm not a Viva employee, and I certainly don't represent in this particular hour my current and past employers. But I think together with Bing, when working in our past post, we have created some compelling story that involved DEL screening and then subsequent medicinal chemistry. And that story can only, I believe, be told as one presentation. And so I was very happy to join him as a scientist to present this story, because I think there are some teaching moments that should be helpful to audience at large. And the story has been recently published in those two JMedChem and also Chem Biochem publications. If you would like to get some further details, I'll go over some of the salient points in parts of my presentation. If you would like to get some further details, you can certainly delve into those two publications. So without further ado, the next slide, please. I'd like to introduce the IDO biology, simply because for two reasons. We will be doing some lead optimization, and so it's important to know what we optimize and for what reason. Also, the setup in the DEL screen requires, of course, deep understanding of biology, and so I'd like to introduce a couple of important concepts here. First of all, it is all about the catabolism of tryptophan. About 90% of mammalian tryptophan is catabolized by those three dioxygenases, namely IDO1, 2, and TDO. They not only differ with amino acid sequences, but also to some degree tissue localization. To be sure, they do share some tissues in terms of where they are expressed, but some of them are relatively unique, such as liver for TDO, which is a tryptophan 2,3-dioxygenase, and immune cell for IDO2, which is a version of course of IDO1. Next slide, please. And so the reaction that IDO1 is catalyzing is this oxidation of L-tryptophan. It goes through an intermediate N-formyl kynurenine, which is then converted to a product of this step, kynurenine, that you see on the right-hand side. That final step is either spontaneous, the N-formyl derivative is not very stable, but also there's an enzyme formamidase that catalyzes that step. And of course, kynurenine is a substrate for the next step in the kynurenine pathway. I will add that this particular step that you see here on the screen is the first step in the kynurenine pathway, and it's the rate limiting step. And so it was always thought to be very important in terms of inhibition. Other steps, maybe you can advance please to the next step, to the next slide. Other steps in the kynurenine pathway, which you see on the left-hand side, are also used for the purpose of drug discovery. But because again, this is the first step was rate limiting, this is a very popular area of intervention for medicinal chemistry. And so why is IDO1 important? And so overexpression and upregulation of IDO1 is associated with neuroinflammatory conditions, and also it suppresses immune function in the body. And so the thought was that by inhibiting IDO1, the variety of diseases could potentially be reversed or controlled. CNS diseases in the upper case, and also in case of cancer, that compound could be a significant addition to a repertoire of anti-cancer drugs. The one concept that I wanted to introduce is this kynurenine tryptophan ratio, K over T, and elevated ratio. And so this is, of course, when the tryptophan is depleted through the catabolism or via IDO1. That elevated ratio has been correlated to Alzheimer's disease and mild cognitive impairment. And it also correlates with increased morbidity and mortality in HIV patients who are otherwise retrovirally suppressed. And so it's an important indication that, again, correlates with a number of disease states. And again, that is the reason why, next slide, please. That is the reason why a number of companies, next slide, please. Number of companies, both large and also biotech companies, jumped into this area in late last decade. Some of the structures of HIV, the IDO1 inhibitors that were developed, you can see on the left-hand side. When GSK became interested in this, not all of those structures were known, but certainly very early entrants into the field were navoximod, which you can see compound number 4 on the left-hand side bottom, and which entered phase 1. And also compound number 2, kind of in the top middle, epacadostat, which went all the way to phase 3. And when GSK entered this field, there was a realization that those molecules are not perfect. They suffered a number of issues. One of them was relatively limited or poor pharmacokinetics, which would necessitate multiple daily dosing. And that was, of course, thought to be not desirable from the standpoint of patient compliance. And so we were seeking new chemotypes in order to address, to basically synthesize, discover better improved molecules. Next slide. And so the effort at GSK went in 2 directions. The first one was the high throughput screen, and I'm not going to discuss much of that. There will be later one slide with some of the key compounds which was discovered in the lead optimization from the hit in that screen, simply because that compound will be very important to understand the mechanism of action of IDO1, which was very unusual. It was different from epacadostat and some of the molecules that I showed you in a previous slide. And it served as basically as a model compound or tool compound rather in this particular case. It just turned out that the DEL compounds went through the same mechanism, and so it was so much easier to understand the DEL compound biochemistry once we understood biochemistry of our high throughput screen compounds. And that will be hopefully apparent in the next few slides that I will address in a few minutes after I give it over to Bing to basically set up the DEL screening campaign in this particular case for the IDO1.
Hey, thank you, Wes. Yeah, hello, I'm back. I realized I speak too fast just now, so I got slow down a bit. Actually, as I teased out a little bit at the very beginning, so that was perfect tool for FIC. But here, I want to remind everybody that DEL is also very critical or very powerful tool for the best in class. Because here we already get many, as Wes said, many hits. But if you need new chemotypes, if you need some new structures, DEL is also a way to go. Okay, so jumping to technology. We did a 4 campaign of IDO1. This is DEL selection conditions. I only have a couple things to highlight in this slide. Number 1, we use a 2 compound, which is a Insight phase 2 oncology compound to help us tease out the real bandits we really want. And in terms of buffer solution, compared to normal ones, we add the aspartic acid and the methane blue is to check out the redox, because there's no ideal one is a redox enzyme. And also I like to point to here a little bit is this DEL selection was conducted at a non-standard 37 degrees. Okay, next slide, please. Okay, so this is data of the DEL selection summary. As you can see, first I got very excited because look at data like this is definitely a DEL ideal ones were DEL friendly target. But in science, in research, the excitement always comes with challenges. So the challenge here is we don't have enough resource to go after every single one. So we have to prioritize. So next slide, please. So to help you to how to read this this chart, actually each square or each rectangle on the is individual library, and the x-axis is the selection conditions for this target or for this campaign is relatively easy, just with and without inhibitors. And the y-axis is the intensity of signal. Here is normalized the enrichment calculation data. As I mentioned, we have so many data and so many chemotypes we could follow up. We have to triage them. So the triage criteria at that time is we pick up the libraries has a signal greater than 4, and also we want to reduce disappeared or reduce signal in presence of the inhibitor. That's kind of like MOA. Then also we want evidence of SAR internal libraries and across library. This is very important. And also at Medcam, Wes maybe know exactly, we want smaller beginning starting points, so that we set up the molecular weight smaller than 450 and CLP smaller than 4. Okay, go ahead, please. Okay, so based on this, we have 9 chemotypes prioritized for the synthesis. We kind of make all of them. Luckily, and 7 of them get confirmed. But today we only have a chance or only have time to laser focus one of them, which is DEL 76. Next, please. Yeah, this DEL 76. Actually, before I go more details of DEL 6, I also want to see when you run the DEL, you need experts to the DEL. You just don't just trash in, trash out. Why we prioritize DEL 76, you have to not only know the DEL, but also know the target. Because as Wes mentioned earlier, the natural substitute of ID1 actually is tryptophan. They have the indole part. That's why we pick the DEL 76 as one of our higher priorities series to be followed up. Next slide, please. Okay, before we check the further detail the data analysis, I'd like to please take a look with this how we make the DEL 76. DEL 76 actually is made, you modify this headpiece, as I mentioned earlier, with this triple bound, acid. Then we do a sonic shear coupling with this about 220 aryl iodines to get this triple bond condition product with cycle 1 BBs. Then this product was subjected to a Larac indole formation to construct this indole ring. As you can see here, of course, it's potential radioisomers on the lower side. Then we hydrolyze the ester, then use 3,000 different functional BBs to capping this acid. We finally get a library with about 3.6 million diversity. That's how we made this DEL 76. Next slide, please. This is a little bit offline discussion. So in DEL community, we have 2 clubs to construct the DEL. This time I show the Viva legal here. Club A is people. It's just this is a thought experiment. We're not done something there yet. So Club A people I don't want names providers. They always promote to form the ring on DNA, like what we did here. Club B is okay. That's too much because the not many reactions can do that on DNA. And number two, maybe not many building blocks can do this to these transformations, then limited the diversity. How about we just pre-made or purchase some core, for example, Indo here, then have some handle ready for degradation. For example, you can do, you have acid here, then you have a lot of building blocks can cap in that 3,000 minimum at least. Those two, not there yet. Sorry about that. Those two methods actually, pros and cons. I mentioned the cons of the club B, club A, but the method B, the molecule generally is relatively floppy. Actually, Viva, we have our own cells. We ask ourselves, can those two clubs meet halfway? Actually, find a way. We already submitted patents. We'll disclose later. Please keep tuned. Later, tease here. Next slide, please. Okay, back to the ideal stories. This time we gonna do a detailed data analysis. Del-76. Normally we use this so-called 3D cube view to do data analysis. Each axis represents the building blocks of each cycle. As you can imagine, a picture, if you get a plane, meaning one building block is very critical. You get a line, meaning the combination of two building blocks is important for the binding. In this case, we find a strong line there, and not so clear, but this line is combination of this blue building blocks and this very special spiral ring green building blocks, then combine, but the building block, cycle one is variables, meaning that's not critical to the binding. So combine all this information put together, we can translate those information to a potential chemotype there. And then normally the way we do the off-DNA resistance, we have three options. We either can do the full wallhead, as you can see there, or simplified version, because cycle ones, pretty much you can use the benzene ring representative or the complete truncated version. As I mentioned before, we already got some series confirmed, so we are super confident with the selection. It has already worked. So we a little bit aggressive here and aggressive and take a risk. We make the truncated version directly. Next slide, please. Yeah. Actually, to our delight, this compound has been confirmed. And also we do a little bit of heat assessment. We can say we can modify that T-beta part to a small part. Because the DEL selection is almost all the compounds already pre-optimized. The quick assessment, meaning it tells us there's not much we can do about this molecule yet. This is a big excitement. We found a very nice hit, but again, excitement always comes with a challenge. Next, please. So actually, we find a super disconnect between the cell-based assay data and the biochemical assay data. So the 7 out of 9 chemotypes were active in cell-based assay, but all of them are inactive in the biochemical assay. As you can see highlighted right here. If one chemotype, so one series is not being confirmed both, it could be coincidence. But all of them, as with such a diverse structure, give us a little bit thinking, maybe indicating something behind has a driving force. Next, please. Even perplexed or confused is actually the target engagement was confirmed with all those biophysical assays, such as CESA, ASMS, PODAN assay. I elaborate a little bit here with the PODAN experiment is we put this compound on a bead, then dealing with the target. Again, that's the one beauty of the DEL. If you get the hit of 2 compounds to this experiment. You have to walk around the molecule to find handle position, at the same time maintain the activity. But for the dial molecule, you can go to the DNA attach point as a handle to make all these experiments. Also, let's go back to a tiny bit the solution or derivatives Viva provided affinity first, because those biophysics data confirmed, and they give us some confidence to go this a little bit further. Next slide, please. So let's take a closer look at the assay conditions. DAL fraction was performed at 37, cell-based was 37, but HTS was performed at room temperature. So what this 12 degrees difference gonna make, but why, and why? I'm gonna leave the question here, let Wes to lead you solve this problem or puzzle. Thank you.
Okay, thank you, Bing. So the question is why, and so this is going to be a subject of next couple of slides, and then I follow up by some of the lead optimization work that I mentioned. And so the insights to this particular dilemma for DEL heat came from that other program that I mentioned, that based on the high throughput screen. Next slide, please. So I wanted to introduce that compound from that other GSK program, compound number 15, also referred to as GSK 5628. So we published on this compound and also epacadostat. We've done some extensive biophysical studies as well as enzyme kinetic studies, trying to characterize and understand both compounds. They are very different in terms of their behavior. They both, at the end, inhibit conversion of tryptophan into kynurenine, and so they are IDO1 inhibitors, but they work very differently. And in the interest of time, I'm not going to go through those details. There are some slides in the supplementary section. If there's an interest and time, we can certainly discuss them. But I will just get directly to the punchline. Next slide, please, and show you what is the kind of the bottom line mechanism that we came up with. And so if you look at the upper portion of the yellow cartoon, this is basically interaction of epacadostat, which is this more classical IDO1 inhibitor from inside with IDO1. And I should mention at this point that IDO1 contains heme in the active site, heme cofactor, and it is very important. It's indispensable in tryptophan oxidation, but epacadostat is capable of binding to the hollow version of IDO1, which is the version with intact heme still bound. And so basically the inhibition is through inhibition of the intact enzyme. Now there is a equilibrium between the yellow hollow IDO1 and the apo IDO1, which is apo IDO1 is the enzyme without heme, and that compound 15 binds precisely to the apo version of IDO1. It doesn't bind to the hollow version; it's too big, but it binds into IDO1 apo version. And when it does, then the equilibrium between hollow and apo gets shifted towards more dissociation of heme from IDO1. And so those are two different mode of the mechanism of actions. In case again of compound 15, it binds the heme-less version of IDO1, and basically the heme has to be removed from the active site in order for inhibitor 15 to act. And that's why it is a slower acting inhibitor. It requires higher temperature, or it requires more incubation time in order to manifest its inhibitory function, simply because there's additional heme dissociation step prior to it. So that explains the data that Bing has shown in previous slide. And we also, as I alluded to before, we also found out that once we understood the mechanism of action, that their compounds went through similar mechanisms, similar to compound 15. And I should add that at the time of our finding, There were others who also confirmed this mechanism, notably some BMS scientists and also scientists from other laboratories confirm this relatively unusual property of ADO1. So now, with the understanding of the mechanism, we were certainly much more confident in terms of progressing those DEL hits. Next slide, please. And so this is DEL hit. It's a novel compound. It has a moderate potency, a cellular potency, 251 nanomoles, and so certainly it needs to be improved. The compound has also relatively high clearance in mouse, which means that it will be relatively quickly cleared from the bloodstream, and so that's not really preferable. And the molecule is also relatively poorly soluble. So clearly, a medicinal chemist had some work to do in order to make those DEL hits viable drug candidates. Next slide, please. And so this slide shows you basically the summary of SAR. Starting from the left-hand side upper box, we first had to examine the chiral nature of this compound. So as discovered, the compound was racemic. It was a mixture of the two enantiomers, and so we synthesized them separately and determined that the 4S isomer was preferred. And then going to the right-hand side upper box, we also determined that the substitution of this fused ring is important, and some heterocycles, such as indazole heterocycle, and so this is when you have nitrogen in place of X, is fully potent, and it actually improves solubility, which is a step in the right direction. But that was not the case for some of the other variants, benzimidazoles, benzotriazoles, and the other isomeric indazole actually lost potency. So after determining this, we moved on to the bottom right area. We tried to substitute the carbamate by amides, sulfonamides, ureas, sulfonylureas. That did not work. There was a loss of potency. So the carbamate was a necessary chemotype, and also small groups such as methyl or ethyl were preferred. And finally, we focused on the left-hand side benzene ring, and so that's the left-hand side bottom box. And there we found out that small lipophilic substituents such as methyl or some of the halogens were improving potency of this compound, whereas any nitrogen in the ring, pyrimidine or pyridines, were detrimental to potency. And before I sort of unveil the lead molecule from this effort, I will add that we only made less than 200 analogs, typically in a lead-up effort leading to a clinical level compound, clinical quality compound. Group synthesize upwards of 2,000 analogs, and that simply illustrates how good the starting DEL hit was. That some of the optimization that Bing has alluded to has already been done at that stage, and much fewer analogs, bespoke analogs, had to be made by medicinal chemists in order to get to the lead stage. Next slide, please. And so this is the lead molecule. It has the indazole group replacement for indole, bromo substituent in the left-hand side aromatic ring, and it is an S isomer and also ethyl carbamate. And as you can see, the PBMC potency indicates that it's about 50-fold more potent than the hit one, and also the clearance values across the species is very satisfactory, with a very low liver blood flow, typically in a low double-digit percent, which is very acceptable. So we felt pretty confident that this compound had lead-like properties. Next slide, please. Certainly, comparison of the rule of some of the rule of 5 parameters suggests that it is a rule of 5 compliant. We also like to use the ligand lipophilicity index as defined by Aztec, the LLE-AT. It is used mostly for fragment evaluation, but it can also be used to basically estimate whether the compound has drug-like properties. And if you look at those values for compound 1 in the table, 0.21 for the hit compound, that's already pretty decent value. So again, this is a reflection of the pre-optimization that is going on in the DEL stage. And then compound 11 features further improvement of that LLE value. 0.29 is close to a good value. Ideally, we would want to have values that are greater than 0.3, but again, this is already quite acceptable. Next slide. And so with that, we embarked on evaluating pharmacokinetics of this compound from oral dosing. I should add here, and this is known, of course, to those who practice in this field, is that high solubility of drug in stomach is very important. If it is not highly soluble, the compound will eliminate as solid, and only a fraction of the drug will actually make it to the bloodstream. Highly soluble compound will be absorbed through the gut wall and then into the bloodstream, and of course, it will be far more efficacious molecule. And so that is what we try to evaluate. Next slide, please. And we initially, don't look at the table yet, we initially looked at the bioavailability of amorphous non-crystalline compound 11, and that compound was highly bioavailable in rat PK. We don't have data in the table. So we felt, pretty confident, and of course, with the crystallized compound 11, and dosed it in rats and dogs in various doses, 5 to 100 milligrams in rats and 5 to 30 milligrams in dogs, as you can see in the table. And disappointingly, the bioavailability F across those species was disappointingly low, single digit by and large. And in fact, the higher the dose, the worse off the bioavailability was. So this was, of course, very disappointing. We also measured passive solubility of this compound, turned out to be very low. And so we thought that we at least knew why that is. This was probably, our hypothesis was that this was due to solubility limited absorption through the gut wall. Next slide, please. And so we embarked on two approaches. We, of course, try to improve the solubility through classical medicinal chemistry. I mentioned before that trying to fudge some heteroatom into the aromatic ring in order to improve the solubility were not really particularly successful. And so that line of work actually didn't yield further advancement. But the prodrug approach that I will briefly discuss here was actually quite successful. Next slide, please. And so in the prodrug, we are, of course, trying to synthesize prodrugs that will be cleaved ideally at the gut wall. And so the drug molecule goes to the gut wall and into the bloodstream, or perhaps they are the prodrug is very shortly lived in the bloodstream itself, and it gets decomposed into a drug and innocuous and quickly cleared promoiety. And if you look at compound 11, there are not that many places that you can prodrug. There is certainly the indazole ring that can be prodrug, but there are two nitrogens, so there is a little bit of an ambiguity. There is an amide bond. You could prodrug that. Those are usually not that very successful. And to make long story short, we considered or we tried many prodrugs. All of them but one turned out to be unsuitable. And the one that actually worked very well, next slide, is this phosphonooxymethyl prodrug 31. And this is similar to the prodrug that is used in fostemsavir. That's the HIV attachment inhibitor from ViiV. And so it works very well for this particular case as well. And this compound is highly soluble, more than 1,000 microgram per ml. And when administered orally to rats, it was highly bioavailable. At high dose, it was almost 100% bioavailable. Now, so we suffered second setback here in that the crystallization of compound 31 was unsuccessful. But what compound 31 at least showed is that the high bioavailability is possible here, and there is a rapid and complete conversion into compound 11 in the bloodstream. So you don't see any really of the prodrug. Next slide, please. And so we did salt screen. So this sounds trivial, but it is a substantial effort, and most of the salts didn't work out. They didn't really give us crystalline hit. The one that worked well was the tris salt compound, tris salt of this phosphate, compound number 32. So that compound, again, as you can see in the table, was still highly soluble. And when administered orally to mice and dogs at various doses, including very high doses, it gave us, middle double-digit bioavailabilities. And so we felt pretty confident or pretty happy with these results. And I should add that compound 32 delivers much more of the drug from oral dosing than crystalline compound 11 itself. And so this is a major improvement in terms of the bioavailability. Otherwise, compound 11 would probably not be a viable drug candidate. Next slide, please. And this is sort of a summary of at the end of the road, when you have all the PK data and potency, you want to look at the predicted human dose. And here we want to cover IDO inhibition 90% over 24 hours. And so the modeling suggests that a low dose of about 60 mg, out of which only 35 mg is parent compound, actually the rest is the pro-moiety and the salt, twice a day will actually do the job. And so we felt pretty gratified that this is this was at the time the preclinical quality type molecule. The compound also didn't show any liabilities in the in vitro tox panel. Next slide. This is belief. So I will very quickly just mention, we are rapidly running out of time, so I will just mention that this chemistry is very different from DEL chemistry. It involves some chiral synthesis of the amine. It's a 4-step chemistry in 30% yield, so very high, very efficient chemistry. And then the prodrugging itself into compound 31, there is this ambiguity of nitrogen 1 versus nitrogen 2. So we established through a variety of spectroscopies that it's position 2, and it's very cleanly it's attached to position 2, which is a very unusual position, I should add. So there is some interesting chemistry and also spectral determination that went into this chemistry. And next slide, please. And this is just the proof that compound 11 works just like compound 15. And so when you look at the UV spectra, the epacadostat, that's the blue compound here, you can see that the red band is shifted slightly compared to the black band, which is basically IDO1 without any ligand. And then for red and green, which are respectively compound 15 and compound 11, there is no shift, but there is a decrease in absorbance, which means that that the heme is actually being expelled from the enzyme due to the presence of those inhibitors and the shift from the holo to apo version of the enzyme. So both compounds 11 and 15 act through that second mechanism that I mentioned that requires that heme leaves the active site. And as I said, we have many additional experiments that prove this. Next slide. And I'm not going to go really through the summary here because I showed you already, again, in the interest of time, I showed you that we had initial issues with confirming hit 1 in the enzyme assay, and this is because it works through a different mechanism which requires higher temperatures or more time. But our experience with compound 15 taught us about this new mechanism, and we then launched lead optimization from compound 1, the DEL hit, into compound 32, which is this tris salt of a prodrug. But the two lessons I think that are important for me is that DEL shortened pathway to drug through the pre-optimization, but nonetheless the medchem was needed to create the viable drug candidate. And so this is basically an integral story where the two parts need to be really told together, I think, to understand how they complement each other. And with that, I'm going to give it over to Bing.
Thank you, Wes. Yeah, before I share my insights, I'd like to give another tale of the DEL and other hit ID synergy. So, as a chemist, or especially like Wes, is always want some backup series to feel comfortable. So we go back to the cube view. As you can see, other than the view generated del70, sparing hit, there's another line, enable lines there. So we pull the structure there, then we line up this molecule with the hit molecule in such a certain way. Actually, if know this structure from the neighboring line from del, I will not open that sparing in this way. It's a very surprising way to open the sparing. But with this information from del, we open that, and also find another computational scaffold designed scaffold has shared some similarity or shared some overlapping parts. Then we do a scaffold hopping to put those kind of three series together. We generate another preclinical candidate. So that's definitely another tale of the del and other hit ID synergy. So that's I'm trying to promote again and again here. Next slide, please. I think I already convinced you that del put a huge impact for this project, including identify multiple leads, helped out the MOA. I mentioned that the two compounds, but I think the take-home message is when you get a hit, especially when you get a hit from del, don't treat it as a routine hit. Please put back to the context, because DNA-Encoded Library is such data-rich platform. All those information, all those data may be already hidden there. You just need to figure that out. That's one big take-home message I would like to share with you. So as Wes talked, when you do a del competing, talk to each other, keep everybody at the same page, will shorten the whole process. Next slide, please. I think that's both me and Wes gonna see the acknowledgement of this target. That's definitely a great teamwork. So I don't have time to read every name there. So everybody is highly appreciated. I learned a lot from those guys. I think that's it.